So, hi everyone, my name is Soa. I just turned fifth year postdoctoral fellow in Shanwoo Laboratory at Stanford University. Today, I will present on how to mass produce functional human iPS-derived cardiomyocytes for industrial and clinical applications. As we all know, heart failure is a devastating heart problem worldwide. Among many, one major underlying cause of heart failure is the deficiency of healthy functional cardiomyocytes post-severe heart muscle damage. Heart muscle is characterized by high density of cardiomyocytes. It is reported that human left ventricle has 2 to 4 billion cardiomyocytes. Since mammalian cardiomyocytes quickly lose their proliferative capacity, when the myocardium is injured, the resulting loss of cardiomyocytes are thought to be irreversible. To restore the function of the damaged heart, investigators have sought to compensate for the loss of cardiomyocytes by inducing cell cycle reentry of the survived cardiomyocytes or introducing de novo cardiomyocytes from different sources. The advent of iPSC technology has opened up a new avenue for generating de novo human cardiomyocytes. Leveraging knowledge from heart development, extensive efforts over a decade have culminated into a simple, robust protocol of directed cardiac differentiation of pluripotent stem cells. Since iPSCs can indefinitely self-renew and the efficiency of directed differentiation is remarkable, the number of cardiomyocytes that we can generate is indefinite in theory. However, in practice, this means everyday cell culture for maintaining undifferentiated iPSCs in continuous differentiation. This is practically labor and time intensive and costly. Also, the differentiation suffers from big batch to batch variability. To overcome this practical hurdle, there have been attempts to expand the fully differentiated cardiomyocytes. One method is by modulating Wnt beta-catenin signaling pathway. Canonical Wnt pathway is mediated through beta-catenin, which acts as a co-transcriptional activator of left TCF transcriptional factor. When Wnt is turned off, beta-catenin is sequestered by forming a complex with many complex proteins, including GSK3 beta. And subsequently undergoes proteasomal degradation. On the other hand, when Wnt is on, beta-catenin can remain in unphosphorylated form and can translocate into nucleus and activate the left TCF transcriptional activity for Wnt target genes. For inducing downstream effect of Wnt activation, GSK3 beta inhibition is widely used using small molecules such as cheer. This study led by Buikima, who is a co-first author of our recent stem cell paper, has shown that beta-catenin plays a crucial role in cardiomyocyte cycling in ventricular wall thickening during development. Buikima et al. Have demonstrated using transgenic mouse model, beta-catenin loss of function mouse heart exhibits reduced myocardial thickness as indicated by the white bar right here, and the quantification compared to the control myocardial wall thickness, and also reduction in cell cycling activity as assessed by the Ki-67 positive fraction of cardiomyocytes. Where, in contrast, constitutively active beta-catenin gain of function mouse heart, shown on the bottom row, showed comparable level of ventricular wall thickness compared to the control counterpart, and also cardiomyocyte cycling activity being comparable to the control mouse. This shows beta-catenin is required for cardiomyocyte proliferation and ventricular wall thickening during embryonic stage. Leveraging this knowledge, many groups have tried to drive expansion of iPS-derived cardiomyocytes by activation of Wnt signaling. For example, Titmarsh et al. Have shown that cheer treatment can lead to almost two-fold increase in Ki67 positive fraction of cardiomyocytes. However, the number of cardiomyocyte increase remained below twofold, suggesting incomplete cell cycle progression. This suggests Wnt activation alone is not sufficient to drive expansion of iPS cardiomyocytes. While reviewing many reports, we realized that the treatment of pro-proliferative molecules such as cheer has always been administered to iPS cardiomyocytes that are cultured in very high cell sitting density, either in 2D or in 3D, where cells are making cell-cell contacts. As you may be familiar, contact inhibition of proliferation is a well-known phenomenon, particularly in epithelial biology. Not restricted to epithelial cells, contact inhibition is a highly regulated process that is critical for controlling tissue growth. As cells reach high density in tissue or cell culture, the cells naturally become less sensitive to pro-proliferative signals and slow down or cease proliferation. Based on this, we hypothesized that cell-cell contacts may hamper stimulating proliferation of iPS cardiomyocytes. So this project was initiated by my talented colleague, Dr. Jan Buikima, a former postdoc in our lab and now a physician scientist at Utrecht University in Netherlands. Additionally, I'd like to mention that this work results from extensive collaborative efforts from 37 contributing authors between Stanford, Utrecht University, and University of Amsterdam throughout the project. To investigate whether cell-cell contact plays a role in regulating cardiomyocyte proliferation, we simply used a passaging technique to modulate cell density. So first, iPSCs were differentiated using small molecule-based Wnt modulating protocol, as shown here. And day 12, fully beating cardiomyocytes were subject to 3 different culture conditions. So first, without passaging group, cells remained in the original well of differentiation, which reflects the most dense condition. Second, the cells in dense passaging group underwent enzymatic passaging process without splitting the cells to lower cell density. Lastly, the sparse passaging group cells underwent enzymatic passaging and plated at low cell density. Cells in all 3 conditions received GSK3 beta inhibitor, cheer, which is a pro-proliferative molecule as shown previously. The result was striking to us. Remarkably, only the cells in sparse culture condition, as shown in this green graph, were able to continuously divide, resulting in almost 200-fold increase in cell number, while 2 other groups representing dense culture conditions showed limited expansion capacity, the degree of which is consistent to the previous reports. To further validate that this is not due to the nutrient or growth factor deprivation in dense culture condition, cell number was controlled and cell density was modulated by varying cell attachment surface area. Specifically, in the dense culture group, the same number of cells were plated at a 10-fold decreased surface area compared to the sparse culture group. We show that sparse culture condition led to increasing the fraction of cardiomyocytes in cell cycle as assessed by pan cell cycle marker Ki67, and more specifically, increase in the fraction of mitotic cardiomyocytes as assessed by phosphohistone 3 staining. This indicates that in addition to cheer treatment, removal of cell-cell contact by sparse cell culture is key to stimulate cell cycle activity and proliferation. By combining GSK3 beta inhibition and contact removal, we demonstrated a remarkable efficacy of expansion of fully differentiated iPS cardiomyocytes. The left-hand side image demonstrates beating cardiomyocytes in one 10-centimeter dish can give rise to 12 T150 flasks worth number of cardiomyocytes. Given that one 10-centimeter dish contains roughly 2 to 4 million cardiomyocytes, this can expand up to generating up to 400 to 800 million cells as shown in panel E. We also tested in 4 different iPS line-derived cardiomyocytes, and we show the comparable capacity of proliferation of these cells by this method. This result implicates that the number required to engineer a human left ventricle myocardium can result from only one 6-well plate worth of pure cardiac differentiation. To investigate the underlying molecular mechanisms, we mainly focused on the effect of GSK3 beta inhibition on cardiomyocyte expansion. Single-cell RNA sequencing was conducted on cardiomyocytes treated with cheer and cultured in low cell density condition. And we found that cheer treatment led to activating Wnt target genes, including axin-2 and LEF1. And this was accompanied by a downregulation of key sarcomeric genes, including alpha-actinin encoding gene, MYH7, TNNC1, and TNNI3, and also calcium handling genes, such as ATP2A2 and ryanodine receptor 2 gene. Additionally, the time course qPCR revealed that the downregulation of these key genes persisted while treating with cheer. This suggests that cheer-treated and contact-removed cardiomyocytes are less mature than the control counterpart. To further examine the maturity of cardiomyocytes, Sarcomere organization was evaluated. So Sarcomere is a basic contractile unit of the myocyte, and its essential component, alpha-actinin, and its registration at the Z-disc, as shown right here, is a widely used indicator for cardiomyocyte maturity. So to examine this, day 30 iPS cardiomyocytes were single-cell patterned and cultured for 5 days, followed by harvested for immunostaining of sarcomeric proteins. Image segmentation and quantification of alpha-actinin, which is stained as red here, the result is shown in the panel C, where the proper Z-disc registration of alpha-actinin with respect to the cell long axis is color-coded as red. Also, in panel D, the quantification of the orientation of alpha-actinin is plotted in histogram with respect to the long axis of the cells. So in other words, 90-degree represents the Z-disc organization at the Z-disc. So by this assay, we found that alpha-actinin of cheer-treated cells contain less sarcomeric alpha-actinin, as evident by reduced number of red color in this image, color-coded image compared to the control cells. And also, we additionally show that cheer treatment followed by C59 treatment, which is Wnt inhibition, is sufficient to recover sarcomeric alpha-actinin organization at the Z-disc. In agreement of immunostaining data, contractility of cheer-treated iPS cardiomyocytes was also reduced compared to the DMSO-treated cells. I hope you can appreciate from this video. And the quantification of the contractile force based on the pixel displacement and also the contractile velocity is shown in this plot. While we have not fully elucidated the contact inhibition mechanism in this paper, one signaling pathway that we have been investigating is Hippo-YAP signaling. So Hippo-YAP signaling pathway is a well-studied, developmentally conserved signaling pathway that modulates tissue growth and organ size control. It is known to mediate contact inhibition of proliferation. Canonical Hippo pathway consists of a signaling cascade of kinases, including MST1-2 and LATS1-2, which ultimately modulates the phosphorylation of YAB, which is a nuclear effector of transcription factor TEAD, that subsequently activates growth-promoting genes. So therefore, nuclear localization of YAB has often been used as a measure of the activity of Hippo pathway. And involvement of Hippo-YAP signaling in contact inhibition of proliferation have been widely studied in epithelial context, and it has been shown that nuclear translocation, as shown in this immunostaining image, demonstrate that the nuclear translocation of YAB is enhanced in low cell density culture compared to the dense. And also it was shown that the nuclear YAB activity is responsible for cell proliferation. In agreement with these previous reports, using immunostaining, we confirmed that nuclear localization is significantly enhanced in sparse culture condition. So to examine whether YAB activity is responsible for increased cell proliferation in sparse culture cheer-treated cells, nuclear YAB translocation was pharmacologically inhibited using a YAB inhibitor, verteporfin. Immunostaining of YAB showed YAB nuclear translocation was significantly reduced when 10 micromolar verteporfin was treated, as evident by exclusion of nuclear YAB in this immunostained image compared to the DMSO treated cells. And the quantification is shown here. Since nuclear exclusion of YAB is known to result in decreasing cell proliferation, we were surprised to find out that the proliferation of cardiomyocyte upon verteporfin treatment did not decrease, but rather significantly increased despite the reduced nuclear YAB localization. Taken together, our data suggests that synergy between GSK3 beta inhibition by cheer treatment and contact removal to induce iPS cardiomyocyte proliferation is independent from YAB signaling activity. Then why YAB signaling is independent in this context? One possible explanation is that Wnt activation via GSK3 beta inhibition may have overwritten YAB signaling. Hillen et al. From Jim Martin group, who is a world-leading group studying a cardiomyocyte proliferation and heart regeneration, have shown that YAB regulates cardiomyocyte proliferation signal through beta-catenin. As shown here, conditional knockout of Hippo kinase scaffolding protein Salvador leads to increase in mitotic cardiomyocytes and overgrowth of heart muscle. but the phenotype is completely abrogated when the beta-catenin is knocked out, indicating when signaling is required for YAP-mediated cardiomyocyte proliferation. Since beta-catenin activity is maximally activated by cheer treatment in our study, the reduction in nuclear YAP induced by dense cell culture condition may have been overwritten. So currently, we're actively working to elucidate the underlying mechanism of contact inhibition. Lastly, we demonstrated the feasibility of using the expanded iPS cardiomyocytes for tissue engineering application. As a proof of concept, the expanded cardiomyocytes over 4 passages for about a month have been encapsulated in Matrigel fibrin hydrogel casted between 2 silicone posts for exerting passive stretching force for augment tissue formation, and cultured for additional 25 days. Then pixel, so the tissue, engineered tissue is shown in this photograph. Then the pixel displacement-based contractility assay was conducted to examine the contractility of the engineered cardiac tissue. We demonstrated that engineered heart tissue made from the expanded iPS cardiomyocytes exhibit the comparable level of force generation, if not slightly higher, compared to those made from their unexpanded, age-matched counterpart. So in summary, we demonstrated iPS-derived cardiomyocyte proliferation is cell density dependent. When activation and maintaining reduced cell-cell contacts enable massive expansion of iPS cardiomyocytes. We show that when activation suppresses maturation of iPS cardiomyocytes. And furthermore, we showed when YAP signaling is not responsible for contact removed induced stimulation of cell cycle. And we demonstrated the applicability of this expansion strategy by engineering cardiac tissue as a proof of concept. And we believe that this can be extended to any other applications that require large quantity of cardiomyocytes. So with that, thanks to all of you for your time and attention to hearing my talk today. And I'd like to acknowledge our fantastic team in Shangwu Lab, as well as all the collaborators who have helped me and contributed to this work. And with that, I'm happy to take any questions.